Welcome to our lecture on cellular organelles. So this is our basic animal cell surrounded by the plasma membrane, and we talked about the function and structure of the plasma membrane. It is a phospholipid bilayer. Contained within the plasma membrane, we have the cytosol, which is the liquid component of the cell. It's where the organelles are in the cytoplasm. Lysosomes are going to be like our stomachs. So they are the organelles that contain digestive enzymes that can help to break down and recycle materials that are taken into the cell via endocytosis, as we talked about earlier also. Here is an exocytotic vesicle. That's using a vesicle active transport to release things that are made in the cell out of the cell. Our Golgi apparatus is going to be where our proteins and molecules that are made within the cell are actually packaged and processed and modified. And so let's start in the center here with our nucleus. So the nucleolus, this is where our ribosomes are made. So remember, the ribosomes are the cell component that takes place out in the cytoplasm. It makes the ribosomes, and the ribosomes join with the messenger RNA, and that is where protein synthesis takes place. The translation of protein synthesis takes place to make our proteins. So it translates our messenger RNAs back to proteins. The nucleolus is contained within the nucleus. The nucleus is our command center. Remember, this is where our chromatin is contained. The chromatin has our genes, and remember, genes are what code for those proteins that are translated into proteins by the ribosomes that are made in the nucleolus. Surrounding the nucleus is our rough endoplasmic reticulum, and you can tell it is the rough endoplasmic reticulum because it is studded with these little red dots, which are our ribosomes that were made in the nucleolus. The ribosomes are what translate messenger RNA into proteins. Once those proteins are made here in the rough endoplasmic reticulum, then they are moved via vesicles into the Golgi apparatus, where, as I mentioned just previously, that they are modified, packaged, and transported through the Golgi, and then they are moved out of the cell via vesicles, via going to exocytosis. They might become lysosomes or become part of the plasma membrane to put more channel proteins or carrier proteins or change the composition of the plasma membrane. So this that we just talked about is also known as our endomembrane system, and we will see that again in just a minute. Mitochondria are the powerhouses of the cell, and that is where our cellular respiration takes place. Recall that you can take glucose and fats and turn them into ATP, which is the energy currency of the cell, via oxidative cellular respiration in our mitochondria. So this is the powerhouse. This is our manufacturing line over here with the command center in the nucleus. And so you can think of our cells as little cities. And here's our garbage disposal, the lysosomes that have those digestive enzymes that were made by the rough endoplasmic reticulum, packaged and turned into the organelle, the lysosome. Peroxisomes are actually for detoxification. So this is where our free radicals will go to be destroyed and to be neutralized. Here is a vesicle being taken into the cell. Remember, that is endocytosis. Our centrosome here, these are what form the mitotic spindle that we saw back when we were talking about cell cycle. And so the centrosomes form the mitotic spindle. They also will be part of our flagellated cell, the only one in the human body, the sperm. They are what form the tail of the sperm and allow it to wiggle and jiggle and move the sperm where it needs to go. And last but not least, we have the smooth endoplasmic reticulum. It also plays a role in detoxification. It also makes our lipids and carbohydrates, and you'll find when we get to the muscle cell that the smooth endoplasmic reticulum in muscle cells is used for calcium storage. You can also have free ribosomes floating out in the cytoplasm. So that is a tour of our cell. Let's go through these next couple slides and look at details. So again, remember our plasma membrane made up of a phospholipid bilayer has proteins embedded in it that carry out various functions such as channel proteins, which allow things to move through, especially those hydrophilic, water-loving molecules will move through a channel. We have receptors, we have enzymes. Proteins are identifier markers for cells. Look back in the previous part of chapter 3 to see all the roles that proteins play in our cells. As mentioned before, our peroxisomes are a vesicular organelle in the cell used to carry out detoxification. So it helps to oxidize toxic chemicals that are waste products of our metabolic processes before they can actually damage our cells. You have catabolic reactions. And so these detoxification reactions are catabolic reactions that help break things down. Ribosomes, we talked about back with protein synthesis. They are bound to the membrane, but they can also be free floating, and they are responsible for translating our messenger RNA into proteins. And remember, messenger RNA was made via transcription in the nucleus from the genes. And remember, genes are segments of DNA that code for proteins. The endomembrane system, I pointed that out when I was going over the cell components, and is composed of our plasma membrane, nuclear envelope, endoplasmic reticulum, rough and smooth endoplasmic reticulum, the Golgi, and the lysosome. So this is kind of our manufacturing in the cell, with the nucleus being the command center. Our two types of endoplasmic reticulum, and make sure you spell these out when you're labeling them, would be our rough endoplasmic reticulum and our smooth endoplasmic reticulum. Rough is where proteins are made. Smooth is where we get carbohydrates, lipids, and others of our macromolecules made. Here is a blow-up and some photomicrographs of both the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The Golgi is where we package, process, and ship those things that are made both in the rough and the smooth endoplasmic reticulum. Here is a photomicrograph of our Golgi apparatus. With the lysosomes, remember these are our garbage keepers. They are full of digestive enzymes that allow the cell to break down dead and dying organelles. Anything that is brought in via phagocytosis, the lysosome can fuse with that phagosome and form a phagosome. Form with the phagocytic molecule that is brought in, forming a phagosome, and then it breaks down and releases via exocytosis. Remember, when you pair them all together, that is called transcytosis. And here is that endomembrane system, starting with the molecules being made in the endoplasmic reticulum, being packaged, processed, and modified within the endoplasmic reticulum. Those vesicles then can go and form lysosomes, which again fuse with things that are brought in, forming endosomes that are broken down. They can become part of the cell membrane to add in more of the proteins that we need, either receptors, channel proteins, markers, anything that the cell needs to function. And then we can secrete molecules out, so your digestive system, all the enzymes that are needed are made within a cell and then secreted out of the cell via exocytosis into the digestive system. So that is a summary of our cytoplasmic organelles. We're going to look at our cytoskeleton quickly. And so the cytoskeleton has three main filaments. We have the microfilaments. Actin is the predominant one of these, helps to support the plasma membrane, form microvilli, and also in cell motion, so with the cilia, that wave-like motion that you can see to move things through the body. Where you'll find cilia in the human body, predominantly in the respiratory and reproductive tracts. They help to form a framework, so our intermediate filaments, you can see them scattered throughout, form a framework of the cell, give it support and shape, provide the ability of the cell to withstand any mechanical stresses. And then last but not least are our microtubules. They form the highway of our cell. So organelles just don't float willy-nilly throughout the cell. They actually are maintained and positioned by little guys. They're called little Dyson, and they carry the organelles along the cell, along this microtubule highway, also associated with the core of our cilia and flagella. And so without this, you couldn't move the vesicles and organelles throughout the cell. And back with forming those vesicles, so the pinching of the membrane to form our endocytotic vesicles, that is carried out by these actin filaments. And as I mentioned before, our microtubules will actually form the core in this 9-2 formation of the base of the sperm's tail. The microvilli, these are going to be finger-like extensions of the plasma membrane. These help to increase surface area, so you will find these in the digestive tract where you have lots of absorption. And then the flagella, as I mentioned before, of the tail, formed by that centriole and filled with those microtubules, and our cilia in the respiratory tract and in the reproductive tract, that's where you find cilia in the human body, form these wave-like motions that help to move things along or create a current within that structure. And here you can see the formation, that 9 2 pairings, 1, 2, 3, 4, 5, 6, 7, 8, 9, that form the center of our cilia and the base of our flagellal tail. And last but not least, our command center, the nucleus, surrounded by our nuclear envelope with nuclear pores in it so that things can move in and out. Big thing we've talked about moving out of the nucleus is that messenger RNA that is made by transcription from the genes of the DNA, and that allows that messenger RNA to move out and then to pair with a ribosome where it can be translated into a protein. All of our chromatin is contained within here, and remember, chromatin is uncondensed DNA, and then when it is condensed, it is considered a chromosome. And here you can see a blow up. Our nuclear envelope is also a phospholipid bilayer, just like our plasma membrane, with these nuclear pores, almost like our channel proteins that we see in the plasma membrane. And then here is that chromatin being condensed into chromosomes by being wrapped around histone proteins, forming a nucleosome, so that we see these classical chromosomes that we think of when we think of the human genome. And then the nucleoli is where our ribosomes are assembled. And that nucleoli, nucleolus, make sure you spell it right. Nucleus is spelled totally different than nucleolus, is contained within the nucleus. And that'll be it for our cell structure. Please let me know if you have any questions. Have a great day.